Hello everyone, I'm Jayden and here is our DNA replication stop motion video. DNA replication is the process of creating an identical copy of a cell's DNA, which would then undergo mitosis and split into two genetically identical daughter cells. This process involves enzymes such as DNA polymerase, helicase, DNA ligase, and RNA primase. DNA, a nucleotide, is composed of deoxyribose, a phosphate group, and a nitrogenous base. During DNA replication, we use adenine, thymine, guanine, and cytosine. Guanine pairs with cytosine, and for DNA, adenine pairs with thymine, but for RNA, adenine pairs with uracil. A cell must pass the G1 checkpoint before it can undergo DNA replication in the synthesis phase of the cell cycle. In DNA replication, new DNA strands are built from 5' to 3'. The first enzyme used in DNA replication is helicase, which breaks the hydrogen bonds connecting base pairs in DNA. Helicase starts breaking base pairs at a location called the origin of replication. As helicase breaks up the base pairs, a replication fork is created. Next, RNA primase adds an RNA primer that's complementary to the parent strand. Then DNA polymerase builds off the primer in the 5' to 3' direction. In this case, the DNA polymerase is following helicase, so this strand is called the leading strand. Then, a different DNA polymerase replaces the RNA primer with DNA. Finally, ligase repairs any nicks and connects all segments of the DNA strand, creating the daughter strand. While this is happening on the leading strand, the lagging strand is undergoing a similar process. However, since DNA is built from 5' to 3', DNA polymerase builds away from helicase after the primer is placed by RNA primase. So RNA primase has to add multiple RNA primers to the lagging strand, and multiple DNA polymerases are used to build DNA segments. The segments of RNA primer are called Okazaki fragments, which are replaced with DNA by a different DNA polymerase. Finally, ligase repairs the nicks and connects the DNA segments together, forming the daughter strand. Overall, the original DNA strand split into two parent strands that became the template for DNA replication. Thank you for watching.